So with this lesson, we're going to be looking at other types of inheritance. So the ones that we've been looking at before dealt with your simple types of genetics that involved a dominant and a recessive trait. With this type of inheritance, it's going to be a little bit different. And we're going to be talking about all the different types of inheritance. So with Mendel's principles, it basically offers a set of rules with which to predict various patterns of inheritance. There's exceptions to every rules and exceptions to the exceptions. So what happens if one allele is not completely dominant over another? What if a gene has several alleles? So basically, despite the importance of Mendel's works, there are important exceptions to most of his principles. In most organisms, genetics is more complicated because the majority of genes have more than two alleles. In addition, many important traits are controlled by more than one gene. Mendel's principles alone cannot predict traits that are controlled by multiple alleles or multiple genes. So the first type of inheritance that does not follow Mendel's principles is called incomplete dominance. So with incomplete dominance, it's basically going to be a mixture. So for example, if we had a red plant, that's going to be, so if we had a red plant, okay, crossed with a white plant, the result is going to be pink. Okay, so it's not going to be red, it's not going to be white, but instead it's going to be a mixture of the two. If this was Mendel's principles, then it would either be red or it would be white, depending on which one was dominant and which one was recessive. So basically, with incomplete dominant, it's a cross between two 4 o'clock plant shows a common exception to Mendel's principles. The efferent generation produced by the cross between the red flowers and white flower plant consists of pink flower plant, as shown. So in this case, neither allele is dominant. Cases in which one allele is not completely dominant over another is called incomplete dominance. So in incomplete dominance, the heterozygous phenotype lies somewhere between the two homozygous phenotypes. So you're basically going to have kind of like a range. Okay, and then you're going to have on one end of the range, it's going to be, for example, the red, and the other side of the range is going to be, for example, just the white. And then basically somewhere in between is going to lie that heterozygous combination. All right, so the only time it's going to be red if it's going to be homozygous for red. Okay, so two big R's. The only time it's going to be white is if it's going to be two W's, so homozygous. And then whenever it's going to be heterozygous, it's going to actually be that blending or that mixture between the red homozygous and the white homozygous. So now the other example is going to be codominance. So we're still looking at that heterozygous, and instead of heterozygous being dominant or dominant, taking on the dominant allele, it's going to represent something else. So with codominance, this is cases in which the phenotypes produced by both alleles are clearly expressed. So this is called codominance. So for this example, in comparison to incomplete dominance, you're going to have your red, okay? And then you're also going to have your white. But whenever we have that heterozygous, it's going to be both red and white. Okay, so both of them are going to be equally expressed. So for incomplete dominance, it's going to be that blending. And then with codominance, they're both going to equally be expressed. So for example, if we had a black cat, right, so big B, crossed with, let's see here. So if we had a black cat big B, crossed with a white cat, WW, we're going to end up with BW. Okay, and this BW is not going to be whatever is going to be dominant. It's instead going to be both black and white. Okay, so that's going to be codominance. All right, so a review. Incomplete dominance is going to be more of a mixture, and codominance is going to be more of that blending where both of those alleles are going to be expressed. Now with multiple alleles, so you're not going to have any examples of, actually you will, you're going to have the blood type example. You're actually going to have a worksheet on blood types. So with multiple alleles, it's going to be a gene with more than 2 alleles is said to have multiple alleles. So many genes have multiple alleles, including genes for blood type. So I'm going to list down, this is going to help you with the worksheet that you have next, all the different blood types. So we're going to have type A blood. Let's move this page down a little bit. We're going to have type A blood. And with A blood, the genotypes for type A blood is either going to be AO or AA. Okay? Then we're going to have, and you should probably write this down, this is going to help you with the worksheet that we have coming up. We're going to have B blood, and it can either be BO or BB. Okay, the next type of blood we're going to have is type AB blood. Okay, and then for AB blood is obviously just going to be AB as the option. And then the last option is going to be type O blood. So type O blood, there's only one option. It's going to be O and O. Okay, so again, for type A blood, we have AO and AA. Type B blood, we have BO and BB. And then type AB blood, we have AB as a genotype. And then for type O blood, we have OO. Okay, so this is going to help you quite a bit. As you can see, there's multiple options, multiple alleles to express blood type. So this is going to be a great example of multiple alleles. The next example, so this is the one that you're not going to see as a Punnett square problem, but you're going to definitely have to know the definition of it. Okay, so polygenic traits are going to be more of a like a wide range of genes. So a perfect example of polygenic traits are going to be skin complexion. So if you look around the room, you're going to have lighter skin, darker skin, and kind of in between. And then there's going to be within darker skin, you're going to have a range of darker skin, and lighter skin, you're going to have a wide range of different complexions. So that's going to be example of your polygenic trait. There's going to be just a wide range of traits. So you want to think of it more of traits that are going to be on like a spectrum, right? As you can see here. So it's just more of like a wide range of traits. Okay, so genes are just not genes, okay? So just because you have a particular gene does not mean that it's going to be expressed in you, and it's going to be expressed the same way among different people. The environment has a huge impact on how these genes are going to be developed and how they're going to be controlled. And one example of it is they do lots of studies that involve twins, and they have adopted twins, and they are placed in different environments. So these twins are going to be identical, so they have the exact same genes. And when you place them in different environments, they're going to be a little bit different. They're going to look a little bit different. They're going to sound a little bit different. They're going to have different interests. So just because your genes say that you're going to look a certain way, the environment is going to have a huge influence on how these genes are going to be expressed. So in other words, the environment in which organisms develop influences the expressions of their genes for living things like wings. Okay, so these are similar species here, and based on the environment, they look different. So this So these wings here are going to be more camouflaged to its environment versus these wings are going to be camouflaged to their environment. All right, so in order to fly effectively, the body temperature of the western white butterfly needs to be 28 to 40 degrees Celsius. The more pigmentation allows a butterfly to reach warm temperatures faster. So similarly, in hot summer months, less pigmentation prevents the butterflies from overheating. All right. Okay, so now you guys are going to work on the worksheets that's going to be handed out, and then ask questions if you need any clarification on how to do these Punnett squares. They're very similar in how you set them up, but it's more in how you interpret them that's going to be a little bit different.